Hello everyone, my name is Ajay Kumar, zoology faculty. Since last 7 years I'm teaching zoology and currently I'm working with the Birla Public School integrated program in Doha, Qatar. Okay. So in today's lecture we'll be discussing about physiology of digestion. What is the meaning of this topic and why we are discussing this topic today? So basically this topic has a very high weightage when it comes to the questions of NEET exam. Majority like 70-80% of questions were asked on this particular topic. And what is the physiology of digestion? So basically the food which we are eating, how it will get digested into different parts of alimentary canal. So this is what we are going to study. So let's talk about the food. The food which we are eating is composed of complex molecules, and there are three complex molecules just like carbohydrates, proteins and fats. So carbohydrate, protein, fats, these are three major complex food materials which we are eating in our diet. And these complex food materials, they are complex and they will be converted into simple substances so that they can be absorbed by our alimentary canal. So this is what physiology of digestion is. So now we'll be discussing about digestion in mouth. In this particular lecture, we'll mainly focus on chemical digestion. Okay, so in digestion of mouth, what is produced in mouth? Saliva. And what is the pH of saliva? 6.8 is the pH of saliva. Is there any enzyme which is present in saliva? Yes, there is one enzyme which is produced in saliva, that is salivary amylase. Okay. Another name of this enzyme is tyrol. Basically, salivary amylase is produced from parotid gland. And the parotid gland is the largest salivary gland. So let's discuss about what is the function of salivary amylase, how the salivary amylase act on our food. There are proteins in our diet, there are fats in our diet, there are carbohydrates. Okay. So out of these three, which substance is digested by salivary amylase? See, there is no digestion of protein or fat happen in our mouth, in our buccal cavity. Basically, digestion of carbohydrate starts in our buccal cavity. Okay, what is the most common form of carbohydrate which we eat? What is the most common? So the most common carbohydrate which we eat in our diet is starch. What are the sources of starch? Starch, there are many sources of starch, just like potato, rice, chapati. So basically, this starch is getting converted into maltose with the help of amylase. pH is 6.8. What is the taste of starch? It's tasteless. And what is the taste of maltose? It's sweet. So when we keep a piece of bread in our mouth, After some time it tastes sweeter because starch is getting converted into maltose under the influence of amylase. Now there is one statement which is written in NCRT. In NCRT it's written 30% of starch is hydrolyzed into maltose by amylase in buccal cavity. Actually 5% of starch is hydrolyzed into maltose in our buccal cavity. But when it start acting, suppose we are eating a piece of bread and the bread is like saliva start acting on it and start digesting it. Okay. After some time it reaches to esophagus and after some time it reaches to stomach. Till that time this amylase is inactivated by gastric juices. In entire duration, approximately 30% of starch is hydrolyzed. Okay, generally only 5% hydrolysis of starch happens in buccal cavity. Okay, so next is digestion in stomach. Now food reaches to the stomach and in stomach there is gastric juice. And this gastric juice is produced by gastric glands. Actually, gastric glands are not such a gland like we can find out, okay, see, this is the gastric gland. Actually, what happens? Gastric glands are not organized glands just like salivary gland, liver, pancreas. They are basically clusters of cells which are scattered in stomach. Okay, so basically, gastric glands, in gastric gland, basically 3 cells are present: oxyntic cells, another name for oxyntic cell is parietal cells, then peptic cells, which are also called as chief cells, and mucus neck cells. So basically these are the 3 major cells of gastric gland. What is the function of oxyntic cells? As we all know, in stomach, highly acidic environment is there. The pH is highly acidic, approximately 2. Okay. So why the pH inside the stomach is highly acidic? Because of presence of hydrochloric acid. So hydrochloric acid is produced by oxyntic cells. Okay. There is one more thing. There is a protein which is produced from oxyntic cell that is called as Cassell's intrinsic factor, or in NCRT it's written intrinsic factor. What is the significance of this intrinsic factor? It helps in absorption of vitamin B12. So this is intrinsic factor. So these 2 things are produced by oxyntic cells. Now the peptic cells. These are the cells which produces enzymes. These are also called as chief cells. So they are the cells which produce mainly enzymes. Enzymes, which enzyme? Pepsinogen, prorenin, and gastric lipase. So basically, I'm using the term proenzymes here. These enzymes are inactive. Inactive enzymes are called as proenzymes. When we put these proenzymes on some food material, they won't be able to digest it. So now these proenzymes are activated into active enzymes. So there are proenzymes and they will be converted into active enzymes. Who convert them? Hydrochloric acid. So this is the most important function of hydrochloric acid, that it converts proenzymes into active enzymes. Okay. One more thing, the hydrochloric acid, it destroys the harmful bacteria, or we can use the term pathogens, of in our food. So these are two important functions of hydrochloric acid. Another function is it changes the pH from normal, like when the food which we are eating, it has a pH of 6.8 or 7, and now when the food reaches to stomach, the pH is converted into approximately 2, okay, due to presence of hydrochloric acid. So it changes the pH to acidic. Okay. Let's study about the proenzymes which are produced. Pepsinogen. It is converted into pepsin. The pepsinogen is an inactive enzyme. It is converted into active pepsin by hydrochloric acid. There is one more proenzyme which is called as prorenin. It is converted into renin by hydrochloric acid. So pepsinogen is converted into pepsin, prorenin is converted into renin. So these two enzymes, pepsinogen is the main proteolytic enzyme of our stomach, and prorenin is a enzyme which is an enzyme which is present in infants. Okay, so these are two enzymes which are produced. One more enzyme, that is the active. This is already active, which is called as gastric lipase. All three enzymes, they are produced by chief cells. Okay. Now the third cell which we are going to study, mucous neck cell. Okay, hydrochloric acid is acidic, so there is a possibility that it can destroy the inner wall of stomach. So the protection from HCl is done by mucous neck cell, which produces mucus. And what is the function of mucus is protection from hydrochloric acid. So this is what gastric glands are. let's coming back to the main thing, how digestion will happen in stomach. As we were discussing about carbohydrate, protein and fat. As we know, pH in stomach is only 2. So carbohydrate digestion won't be there. Why? Because it's not possible. The amylase which was there in which was produced by salivary gland is not here, or it's inactive. Now remaining are protein. So the proteins which we had in our diet and the fat. Let's talk about digestion of these two. Okay. Digestion of proteins. These proteins, they are digested by pepsin, HCl was there, pHs. The proteolytic enzyme pepsin, it works in acidic pH only. So these proteins, they are converted into proteoses, peptones and peptides. So what are these things? Basically protein, there was a long chain of proteins and this long chain is broken down into smaller chains. Still they are complex, but they are less complex. So we can say that long peptide or long polypeptide, it is converted into smaller peptide chain. So this is what happened in stomach. Now in infants, there is a milk protein which is called as casein. Under the influence of renin, it is getting converted into calcium paracasein. And this process is called as curdling of milk. So this is what happened in stomach, digestion of protein. Okay, one more thing which is remaining, fats. What happened to fats? Approximately these are the fats. So approximately 1% of fat, they are getting converted into fatty acid plus glycerol. 1% only. The enzyme is gastric lipase. So this is what happened in stomach. Okay. So in mouth, digestion of carbohydrates starts. In stomach, digestion of your protein started. Okay. And slightly small amounts, approximately 1%. Okay. So 1%. So there is one question which can be asked: why only 1% of fat is digested by gastric lipase? Because lipase act on emulsified fats. Okay. So what is emulsification? We'll be studying in next topic. So only 1% fat or small amount of fat is emulsified in this particular particular site in stomach. So that's why only 1% fat is broken. Okay. There is one more question which can be asked. The term is chyme. What is this chyme? The food which is mixed with the secretion of gastric juice is called as chyme. Okay. So food plus gastric juice is called as chyme. What is the pH of chyme? As usual, because it's in the stomach, pH is 2. Okay. Now we are moving to the third point, digestion in small intestine. So digestion of small, how digestion happens in small intestine? See, majority of digestion is happening in small intestine only. Okay. So the food which is coming is called as chyme. Okay. The pH is 2 and it's acidic. Now this food will be converted into chyme, and which is alkaline. How? We'll be discussing this. So in small intestine, secretion of 2 glands will be there. One secretion will come from liver. So from liver, bile juice will be there. And from pancreas, pancreatic juice will be there. Out of these, the question which can be asked is, out of these 3, which one is true digestive juice? Bile or pancreatic juice? So answer is pancreatic juice. Pancreatic juice is true digestive juice. Why? Because it has enzymes. So they can ask you a question like this, or they can ask you why bile juice is called as pseudo digestive juice. Why? Because it has no enzymes. Okay. So this is the difference between these two. Although there are some other differences also. Now, as I told you, chyme is there and it is converted into chyme. Chyme is alkali. So the secretion of liver, which is like bile juice, plus pancreatic juice, they convert this chyme into chyme, the acidic chyme into chyme, and the pH will be 7.8 or okay. So now initially it was neutral. Now when it comes to stomach, it became acidic. Now it became again alkaline. Okay, slightly alkaline we can say. So this is about the pancreatic juice. Now we'll be studying about enzymes. So in pancreatic juice, proteases are there. What are these proteases? Proteases are the enzymes which help in digestion of protein. Okay. Now, proteases are trypsinogen, chymotrypsinogen, and procarboxypeptidase. And all are inactive. Now question will come in your mind. So why all the proteases which are produced, whether they are in stomach or intestine, why they are acidic in nature? Sorry, why they are inactive? Because our cells, see, all the enzymes, they are produced by some cells, na? They are secreted by cells. And cells are also made up of protein. If we release those enzymes in active form, first they start digesting the cell. Okay. So that's why they are released in inactive state. Later they will get activated. So this is about the proteases. Then there are some other enzymes, lipases are there. Then there are amylase is there. Now all the three major compounds, whether they are carbohydrate, protein and fat, they will be digested here. Okay, so they can ask the question, which part of alimentary canal have the digestion, like perform the digestion of all the three major food particles? Answer will be small intestine. So now first topic is digestion of carbohydrate. In small intestine, digestion of carbohydrate. So first one is starch is there. How much? Approximately 70%. So the starch is broken down into maltose with the help of pancreatic amylase. Okay, but maltose is a disaccharide. It means it is still complex. So the maltose is converted into glucose plus glucose by the enzyme maltase. The sugar which we are eating, sucrose, is also digested into glucose plus fructose by enzyme sucrase or invertase. Now the third one, the lactose. Lactose is a natural sugar which is present in milk. So the lactose is converted into glucose plus galactose. So these end products, whether they are glucose, fructose or galactose, all are simple one. It means they can be absorbed by our intestine. Okay, so this is how digestion of carbohydrate completes in intestine, basically small intestine. Now let's talk about digestion of proteins. So the digestion of protein, it started in stomach and there were peptides. Peptones and proteases. They were formed. Okay. Now we'll be digesting these. But to digest them, first we need to activate the proenzyme. So to activate the proenzymes, first trypsin. The trypsinogen is activated by enzyme enterokinase. What is this enterokinase? This is also an enzyme which is produced in small intestine, okay, and it helps in activation of trypsin. Now this activated trypsin helps in activation of other proteases, just like chymotrypsinogen, into chymotrypsin. Activated by trypsin. There is one more enzyme which was there in pancreatic juice. So that is carboxy peptidase. So carboxypeptidase is the enzyme which is also converted into active state from inactive. So inactive carboxypeptidase is converted into carboxypeptidase by trypsin. Okay, so this is also activated. Now we are moving to the next part. These are peptides. Proteases and peptones. They will be converted into dipeptides. Under the influence of trypsin, chymotrypsin, and carboxypeptidase. Still, it's a disaccharide. Okay. Now this dipeptide What is this dipeptide? Di means two, peptide. The peptide which has two amino acids. Okay, so there are two amino acids which are linked by the peptide linkage. So to digest this dipeptide into amino acid, there is an enzyme which is called as dipeptidase. And it is produced by intestine. Okay, so it is produced by intestinal juice. Succus entericus is the intestine juice. Now moving to the next part, digestion of fat. In the digestion of fat, the one enzyme which is there, that is the lipase. But there is one problem with lipase. This lipase is unable to digest the unemulsified fat. See, there is one molecule, one large molecule of fat. This is the large molecule of fat. Now, this lipase won't be able to digest it because there is no surface area for attachment of this lipase in this large fat molecule. Now this large fat molecule is broken down. This large fat molecule is broken down into smaller units. And this process is called as emulsification. And this process is done by bile juice, bile salts. So these are the two main functions of bile juice. First thing, it converts pH from acidic to alkaline. Okay, and second function is emulsification of fat. So now these fat molecules, they can be digested by lipase. So fat. The fats which we are having, they are triglycerides. Triglyceride means there is one glycerol molecule, and along with the glycerol molecule, three long chains of fatty acids are there. So this is the meaning of triglyceride. So this triglyceride is converted into diglyceride plus fatty acid. So this was the chain, and now it is one fatty acid chain is removed. So this is how digestion of fats happen. Now this diglyceride will be converted into monoglyceride plus fatty acid second chain is also removed. Now this monoglyceride is also broken down into fatty acid plus glycerol. And all this process is catalyzed by one enzyme only, name is lipase. This is how digestion of fat happens. Are we missing something? Do we eat protein, fat and carbohydrate only? No, there is one more thing. We eat nucleic acids also. So what are you saying? Nucleic acids. How we are eating the nucleic acid? Just like how we are eating the DNA and RNA. All the cells are having DNA and RNA, whether you are eating meat or you are eating vegetables. They are made up of cells and they have DNA and RNA. And DNA and RNA are also complex molecules. So we need to convert these molecules into simple one also. So DNA and RNA, they are called as polynucleotide. Okay. So for DNA and RNA we have the enzymes which are called as DNase and RNase. So these DNases and RNases, they convert these polynucleotide into nucleotide. Okay, that's it. Nucleotide is finished. Is it a simple molecule? No, it's also complex. A nucleotide is also made up of three things. This is a pentose sugar. There is one nitrogenous base and there is one phosphate. So this is a general structure of a nucleotide. So still it's made up of three things. So we need to break down these three also. So the nucleotide is also broken down into nucleoside plus phosphate. So this phosphate, this bond is broken down, phosphodiester bond is broken down. So now this phosphate is there, remaining structure is nucleoside. The nucleoside is converted into sugar, which is pentose, plus nitrogenous base, and this is nucleosidase. The enzyme is nucleoside. So this is how digestion of all these things happen in small intestine. Now digestion is over. So these monomers, they will be absorbed by small intestine. So digestion happen in large. Is there any kind of digestion which is happening in large intestine? No, there is no digestion which happen in large intestine. So that's all about this particular topic in the physiology of digestion. In this particular topic, multiple questions can